Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Hello, this is Richard Jacobs with the Finding Genius Podcast. My guest today is Assistant Professor Moses Bility. He's an assistant professor, infectious diseases and microbiology at University of Pittsburgh. I'm going to talk about some of the dynamics of HIV and AIDS. So, Moses, thanks for coming.
Thank you.
Yeah, tell me a little bit about your background. Why do you work in the area you work in, first of all?
Yeah, so my research sort of inspiration is really, sort of triggered by, work from, Stephen Hawkins and Leonard Maladona. And really sort of what they sort of argue is that we need to, reevaluate models that we have for physical phenomena. And it's sort of really sort of that background that I sort of move into infectious diseases, really trying to develop model system that recapitulate human diseases, and also looking at some of the foundational paradigm that we have that govern those models that we develop to explain and predict the dynamics of infectious diseases.
When you say recapitulate human diseases, meaning cause them to happen in a similar way in a mouse, but without having to, be unethical and infect someone and watch the disease progress in them.
Correct. Yes. So there are differences in between, a rodent and a human being. For some viruses, they don't infect they don't infect rodents like HIV. And so you have to sort of recapitulate that human organ system in a rodent model, and that will allow you to study the disease. And so that's sort of one aspect of my work. And then the other aspect is sort of looking at some of these foundational paradigms. And so that's more like a theoretical aspect of my work, and how, because a laboratory really doesn't tells you everything that sort of goes on in society. It's just a snapshot, and you're making a lot of assumptions. And what I try to understand in terms of sort of broader infectious disease research is what are some of those things we're missing? So that's sort of the second part of my work. Yeah.
So what are the, to you, what are the most important questions that you're trying to answer with your research right now?
So obviously COVID is on everyone's mind, and including myself. And so one of the things that sort of stands out with this COVID pandemic that I think everyone noticed that is a lot of the early predictions in terms of where the regions that were most at risk, namely in sub-Saharan Africa in the equatorial belt, where you have really poor health system, and also in Southeast Asia, where, for example, Thailand, you have high level of tourists from China, and so that didn't really pan out. So, and it's sort of going back to, Stephen Hawkins' model-dependent realism sort of argument, sort of thinking that I sort of gained from that is that we need to look at these foundational paradigms that sort of governing those hypotheses that were sort of developed. And so that's been another aspect of my work, sort of looking at those dynamics and trying to understand what we're missing.
So do you want to focus on COVID, or do you have a much longer term look into HIV? And I'm sure you've learned a lot of lessons there. So if we take those same questions,
right,
find HIV, which has been rampaging for, 30, 40 years. What's been learned there, especially by you, that's really unusual or different?
So in terms of HIV, so where my work has really sort of focused is on looking at macrophages and iron. And in terms of, because macrophages really, they're sort of these multifunctional cells that play a role in maintaining tissue integrity, that play a role in initiating immune response. And so I've looked at, so I developed this humanized mouse model that has human thymus and spleen and also immune cells. And the idea is that this model recapitulates the human immune system. And we've used this model to sort of look at what are some of the mechanism that allows this virus to persist versus being controlled. And we can use different mutant virus called a NEF virus, NEF mutant virus, to really sort of look at those dynamics. And the really exciting outcome we had in our lab is showing that HIV, NEF mutant HIV can be controlled in this model with the robust human immune system versus wild type virus.
Well, again, what are you trying to figure out in relation to HIV?
So what we're trying to figure out is what are the signaling network that sort of drive that control. And also, what are some of the molecular determinants of the virus that sort of drive that? And right now, we're sort of looking at macrophages, how the virus perturb macrophages, and particularly has to do with sort of the metal status, iron status in these macrophages, how the virus sort of perturbs that. That allows the virus to either persist or the immune system to suppress the virus. And we've been able to.
It seems like a lot of viruses somehow know to attack, our immune cells. I mean, maybe since viruses appear to be passive in the virion phase, that those are the cells that they tend to run into first because they're like landmines, I guess, floating around. And experience has taught them that the immune system will attack them and engulf them and all that. Have you been able to, analyze, or has anyone been able to analyze macrophages mid-phagocytosis and see what's happening to, let's say, HIV?
So there's been some group, we haven't done this specifically. There's some groups sort of looking at sort of the cellular dynamics of the virus and there's particular regards to macrophages. That is something we're interested in doing sort of in the future. But you raised a good point, but a lot of that in terms of targeting, you have to sort of think about that from the perspective of, that cell having that entry receptor. So that plays a huge role where in macrophages, they do have those entry receptors. Versus other viruses, they don't. And so the dynamics is a whole lot different.
So you're saying the iron status of the macrophages, I mean, the only iron use I've ever heard of, and I'm sure most people in the body is, I guess, hemoglobin, red blood cells take it up and use it. Right. How do other cells use iron? I mean, in particular macrophages, what role does it have in them?
So the macrophages, they recycle iron. So most of the iron in your body is sort of recycled iron. You excrete very little iron and you intake very little iron. So the macrophage sort of they're sort of the distributor. They sort of decide who gets what amount of iron, and who doesn't get iron. And they use that in sort of host defense, particularly in terms of bacteria. But you're right, in terms of making, red blood cells, that iron is critical for transporting oxygen to different tissues in the body. So you can sort of see how, on that aspect, how it's critical, but also in terms of restricting bacteria growth. Bacteria in most organisms require iron to grow. In terms of viruses, we started to explore how viruses and iron sort of interact, at least vis-a-vis the macrophages. And my work in sort of looking at COVID sort of moved into that realm, sort of looking at a more bigger picture and sort of understanding how iron, this ferromagnetic metal in human, how does it modulate disease, and how does the environment interact with that iron to affect human health?
Well, why do bacteria preferentially need or use iron to, to spread? I mean, to live, period?
Yeah, so for the replication, DNA synthesis, a lot of enzymes that bacteria use require iron as cofactors. So it becomes really critical for the, bacteria even make molecules that can sequester host iron for their own use. So it becomes really critical to have access to those iron, and the host, in particular the macrophages, whose job is to sort of secure those iron and redistribute them to the host cells. there's sort of a competition between the macrophage and the bacteria. A lot. But in terms of viruses, there's very little research done on that right now. And we're actually one of the few groups that sort of look into that. We started during my postdoc days. I started looking at in terms of hepatitis, viruses, and macrophage and iron, and now I'm moving to HIV. And obviously moving into COVID, sort of looking at what role iron could be playing in these diseases.
Do you look at, let's say, can you induce a low iron state or a high iron state in mice, or model anemia in people? What's a way to model this really easily? Iron disorders?
Yeah, so for a lot of in terms of looking at iron, we sort of looked at a natural interaction. So we infect these animals. So we create these animals with a human organ system, and then we infect them with a virus. Right now we're doing a lot of work with HIV. We've done a lot of work with HIV, and then we see the changes in the iron status. And what we've sort of observed is that this changes in the iron status also change the inflammatory status of these macrophages, and we see more tissue fibrosis. So, and this becomes really critical because tissue fibrosis sort of damages the lymphoid tissue. That's really critical for generating those immune cells that can control a viral replication.
So, how do you?
Yeah,
I've heard of organoids being made and tested in vitro, but how do you change a mouse's physiology to have human organs in it somehow? What are you doing?
So we're doing transplantation. So we transplant these animals with human hematopoietic stem cells. We also put in tissue, sort of organoid-like tissue in the animal. We put it on in a specific sites that sort of really allows the tissue to grow, predominantly in the kidney capsule. And then we sort of investigate whether or not these immune system that develop in these animals sort of recapitulate to what we see in humans. So we look at different cell types, and we look at the responses. We can also couple these with other organ systems, sort of like the liver, and recently we're moving into the skin and the lung. So we can transplant those tissues in there and sort of look, that will allow us to look at the interaction between that human host tissues and that pathogen, that virus in vivo. And the animal is immunodeficient, so that sort of allows us to sort of implant those tissues. So that's a critical aspect. So we're using immunodeficient animals.
Yeah, but again, my liver is bigger than a whole mouse.
Right.
So how do you do a miniature liver? Do you take
So
parts?
So there's an in-grain, sort of biological limits, right? So if you put a human liver cell in a mouse, it tends to grow to that limit, that biological limit of that host. It doesn't grow any bigger than that. So if we put thymus and spleen in there, it doesn't grow beyond the capacity of that host to maintain that tissue and sustain life. So that's kind of fascinating. So what are some of the molecular mechanisms that determines that? Certainly mechanical stressors plays a critical role in determining tissue size. We haven't really investigated that, but you're absolutely right. There's some interesting biology that sort of goes on in terms of maintaining that appropriate tissue size to allow us to study those dynamics. The host.
So if you put in an immunocompromised mouse, if you put some liver cells in there, it'll grow like a tiny human-esque liver inside the mouse.
Right. It'll grow like grow like a tiny human-like liver, and we transplant it so the cells go directly in the liver, and it sort of reconstitute the liver. We can get, depending on the cell type we use, you can get anywhere from 20% to up to 90%, depending on the cell type.
Wait a minute. So this is like this. How good are they, the livers that are grown, for instance, in a mouse? Are they differentiated? They have blood supply?
Yes.
This could be a billion times better than the organoids that are being made just by a little ball of cells.
True. That's the exciting part about this work, in that it's you're recapitulating the organ system in vivo, where you have system to system interaction. And in our case, we're looking at the immune system and the liver cells. We extended that to the skin and the immune system, and now we're moving to the lung. So it's sort of really exciting because it's sort of, moved beyond, that, organoid tissue culture based system that you really don't have that sort of systemic interaction. And there's certainly, a lot of biology that is lost by not looking in vivo.
Yeah, that's amazing. Has that been, I would think this is being studied actively by a lot of people. I mean, if you contrast, I don't know, one of these liver organoids or whatever other organoids to one developed in a mouse, I mean, do they act similarly, differently? Like, how good of an approximation for a person is?
So there's been some studies, not from my lab, but actually looked at the prediction of drug toxicity and drugs that fail in clinical trial, based on the traditional mouse models. And what they found was that these humanized liver mouse predict the toxicity that occur in a clinical trial. And whereas the traditional mouse model didn't predict that toxicity. So with the organoid system, the biggest problem is sort of maintaining that tissue, that cell, that organoid in that tissue culture system. It's really difficult. There seems to be, particularly for the liver, there seems to be that in vivo system. There are certain nutrients, micronutrients, factors that is really critical that it's just almost impossible to sort of, replicate that, to recapitulate that in the tissue culture system.
When a mouse has, liver cells in it, do you take out the mouse's liver or that exists, at the same time that the mouse's liver exists?
Yeah, so we have to do like a gradual depletion of the mouse liver because liver is a critical organ. So we have to do a gradual depletion of the mouse liver cells and reconstitute that with the human liver cells. So that's sort of really critical, sort of managing that. And we sort of really optimize that. So where you knock out the mouse liver cells and then the human cell sort of reconstitute that. That's sort of the overall scheme of that reconstitution.
And how many different, so I guess it's just one organ? Or have you had, poor mice with 2-3 organs that are, that are humanized, that are growing inside of them?
Yeah, that's sort of, that's sort of, the work we do in my lab. We built multi organ system humanized mouse model, and so we've built liver and immune system from autologous tissues. We've moved into the skin, the immune system. And that sort of really allow us to study, for example, skin pathogen and the inflammatory response to that, especially on MRSA. So we have a project on that, and we're also looking at intradermal vaccinations in that. So some of the work with COVID, we're looking at intradermal vaccinations because it overcomes a lot of the restrictions associated with intramuscular vaccination, where you need cold chain. And dosing issues. So by using these skin patch vaccines and taking advantage of the high concentration of antigen-presenting cells in the skin, we can sort of overcome that. And this model we've developed really sort of allow us to sort of test that.
Yeah, I would think all the organoid community would be all over this. I mean, this could be a new standard, for clinical trials, because if you have your mouse model and everything, and then alongside of that you have the mouse with the humanized organ and stuff, I mean, it seems that would probably give you a lot better intelligence about what will fail and what won't going forward. So why not make that a standard? I mean,
right.
I would think the FDA would be all over this.
Right. So there's been considerable interest from the FDA in terms of utilizing these models. There are some limitations of it because one of the sources of tissue that we use and it involves fetal tissue, which is medical waste from elective abortion. So that sort of, there's been some concerns about that. And so that sort of damp the enthusiasm, at least in terms of sort of moving forward with that work. So we don't, so there's been sort of like down, I would say that's sort of the down on that research because there's been some limitation on that. But we were looking at other sources, sort of trying to look at, iPSC, sort of generate the organoid and then transplant it in the animal, and also using tissue from adults, surgical ways. So we're looking at different alternatives. So yeah, there's some hurdle in terms of sourcing, tissue sourcing that sort of have dampened the enthusiasm for this work. But as considered.
What if I understand why, because you could say, it would encourage abortions to get the raw material needed for this. But what happens if you take adult stem cells and you put them in a mouse and you do the same procedure, or, induce pluripotent stem cells? Can you cue it to grow at least a small version of a given organ?
Right. And that's sort of where, that's where the field is moving, and there's more interest in funding those line of work. But one of the fundamental limitations is that tissue stem cells from adults and older individuals just aren't as good as those from fetal-derived donor. So we still need to sort of optimize those, iPSC-derived cells, or adult stem cells. So there needs to be significant optimization of those protocols, and in terms of getting them to comparable levels to what we achieve with the fetal-derived tissues.
Huh. Yeah, at least to me, I think you can hear this is a real interesting part. What are some of the limitations? Can you do a heart inside a mouse, or is that not possible? Or what organs are possible, and how far do you think this can be pushed?
So in terms of the list of the organs, so we've successfully engrafted, hematopoietic stem cells, the lymphoid tissue, the spleen, and the thymus. We've recently coupled that with the skin, and we're sort of moving to couple that with the lung. And we previously, co-transplanted the hematopoietic stem cells and liver progenitor cells. So we've to me, that at least from my research interest, those are some of the major organ systems that we're interested in. But there's obviously a lot of it involves surgery that sort of create a hurdle in terms of the organ systems that you want to transplant in. And we're sort of looking right now. We sort of move into larger rodent species, and so the rat, which really sort of allow us to do a lot more surgical manipulation as opposed to mouse models.
Yeah, if this model worked in a pig, you could probably grow full-size organs, maybe for retransplantation back into people, or if you got like a capybara, like a big rodent,
right,
or a guinea pig. Even a guinea pig, well, it probably wouldn't get you there, but let's say a thyroid, what's the smallest animal that you could do this in that could even grow like even half a human thyroid,
Right. Yeah. So there is some work, and I believe the state of California sponsors some work sort of growing human organ system in pigs for the specific purpose that you just stated, for transplantation. So there is considerable work on that, sort of humanizing a pig for the purpose of transplantation. So I haven't really followed them, in the last few months, but they seem to be making some headways in that effort.
What happens when you grow, again, a liver or a lung or whatever it is in the mouse? It's a lot smaller. Like you said, it's like a fish that grows to the size of a tank, I guess. But what about its morphology? Are you growing literally a tiny lung that looks just like a regular lung, or does it have a different morphology, or liver, or et cetera?
So the lung work, a colleague of mine, Richard Garcia at UNC, he sort of did that, and he really showed that you can sort of recapitulate a lot of the lung architecture and lung structure. We sort of move in that primarily because of COVID. With the skin that we worked on, we show the same microanatomy. We show hair follicles. We show multi-layer epidermis. And so in the liver, we can see the similar structure in the liver. With the lymphoid tissue, we can see similar structure in the lymphoid tissue, the follicles. So really, we were able to recapitulate a lot of the microanatomy and look at the function of those cells in those tissue. We can recapitulate the function of those cells. So to me, that's really exciting because now we can sort of look at how does the human immune system react to HIV? And how does that immune system control the virus? So by using virus that have mutants in these virulent factors, we can see how those virulent factors sort of perturb the immune system to mediate chronic infection. So, and that sort of opened a whole new window for sort of evaluating vaccines, because now you have a human immune system that it can control a live attenuated virus. To me, that's sort of the gold standard for evaluating a vaccine.
Again, when you look at an organ that is much smaller, made in the mouse, you're recapping it. But what's missing, in your opinion? Like, again, are there what does that tell you? If you grow a liver in a mouse, a human liver, it's a lot smaller, but it's not like an exact micro replica of the liver. Like, what's different about it? What's missing?
So it depends on the tissue you're sort of looking at. In liver, because we're putting sort of liver progenitor cells. We can recapitulate a lot of the cell types. If we put in for fetal-derived tissue, we get some of the hepatocytes to differentiate into adult-like state. We can get some stellar cells in there, but the endothelial cells, they seem to be a lot lower in terms of number of cells, and so that's certain areas. With the lymphoid tissue, they're in the kidney capsule, so it's not precisely the environment, even though you have high level of blood flow in the kidney capsule, which you can use to screen out pathogens, but still the environment is a little different. Same with the thymus. So there are some differences in terms of where they're located, but in terms of what I'm interested in is sort of functional, those that function as that. And for the most part, at least this model we've created, this BLTS model, we are able to control a live attenuated virus. And that's the hallmark of a robust immune system, to be able to control a live attenuated virus.
Do you think in the future, even if, let's say someone's kidneys are shot, do you think in the future, even if you were able to implant 3 or 4 mini kidneys grown in a mouse, would that still work? Would that be enough function for the person to at least function at a low level and get along?
Yeah, so that's sort of really interesting. I've never, so my work is really sort of like animal modeling. I've never really sort of, ventured into the realm of sort of human transplantation biology. But there are a lot of groups that are sort of looking at cellular transplantation as a means to sort of, assist and prolong the life of people on the organ transplantation list. So a lot of times it's sort of a race against the clock, and there's been tremendous progress in that area, but there's still a lot of work needs to be done in terms of making sure those cells engraft and those cells can actually grow. In the mouse model that we work in, there might be some advantages because number one, the small size, so we don't need to put a lot of cells in, and these animals are immunodeficient, so we literally knocked out their BT cells and their NK cells. So there's a little bit less restriction in terms of accepting those xenografts. So that might be a major hurdle within humans, wherein you have a human immune system that can reject that xenograft, that donor tissue.
The mice are immunocompromised, but are they germ free?
No,
do they have a microbiome?
So they do. We do a specific. We run a specific pathogen-free program. So just sort of a list of pathogens that we're mainly concerned about because they cause disease. But they're not germ-free. They can't be made germ-free. To sort of look at some of the interaction between the microbiome and the immune system. So that's another exciting area that can certainly sort of looking at how the human microbiome interacts with the human immune system, which most likely is different from, with the interaction between the mouse microbiome and the mouse immune system.
Well, if you grew a certain organ in a mouse and one was germ-free, one was not, you could see if that affects the growth of that, mini organ too.
I agree. Yeah, not just the growth of that organ, but the function of it. The microbiome play a critical role in terms of modulating immune cell development and function. So certainly, you can you're likely to see there are differences in terms of the architecture and the growth and the function of those cells in those different under those different conditions.
Well, very interesting. So, Moses, what do you working hard on that you think in the next 1 to 3 years you're going to make some headway in understanding?
So I'm really sort of like I said, interested in terms of understanding how HIV sort of perturbs the immune system, and really the idea is that information from that, from those experiments, will really sort of give us an idea to design vaccines. And so we've had this really exciting result where we show we can control a live attenuated HIV in our animal model. And we're going to do some machine learning analysis and look at network interactions of the immune cells and try to understand how do we sort of design vaccine based around those immune responses that control those live attenuated virus. Obviously, you can't use live attenuated HIV in people because there's inherent sort of risk with that, issues like, reversion of that virus. But you can use that information that you get in terms of the immune system controlling that virus to design a vaccine that can trigger similar immune responses. And really sort of that's where in the next 3 years, that's where my research is really sort of oriented towards determining.
Very good. Well, Moses, I mean, it's been a great call. I know I kind of took it in one direction, but
that's
really fascinating. So.
No, that's I didn't know which direction you wanted to take it. I was prepared for whatever discussion you wanted to have, but I definitely do appreciate that, and this is important work that I do. It's a major part of my research program, and having this discussion and sort of putting it out there so that people understand. And you make a lot of really good points that these model systems could have tremendous impact in terms of drug testing and vaccine evaluation, and that's really where that's the direction I hope to move into.
When you spare time, I need you to solve HIV. Make it the new standard to use these human little organs in mice, and then solve the transplant problem as well. So can you do all that?
Well, we'll try. We'll try. Definitely, we'll try.
Right on. Well, Moses, thanks for coming. I really appreciate it.
I appreciate your invitation too. Thank you.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.